As you begin to settle into this calm exploration, allow your attention to gently drift away from the large and familiar structures of daily life and slowly move inward toward a quieter scale. Here, far beyond the reach of ordinary sight, exists a world of remarkable order and consistency. It is a world that operates continuously, without rest or awareness, yet supports every breath, every movement, and every moment of living experience. This is the microscopic world of cells, the smallest units capable of sustaining life. Every living organism, from the tallest tree to the smallest insect. In the human body alone, there are trillions of them, each one enclosed within its own delicate boundary, each one carrying out its tasks with steady precision. Cells are not passive building blocks, they are active, dynamic systems. Even as the external world changes from moment to moment, the cell remains remarkably stable, preserving the conditions required for life to continue. To understand how this stability is maintained, it helps to imagine the cell as a small, self-contained space floating within a larger environment. Inside, chemical reactions occur in carefully controlled sequences. Energy is transformed, materials are assembled, signals are interpreted, and waste is quietly removed. None of this could happen without a clear separation between what belongs inside the cell and what remains outside. This separation is neither rigid nor absolute. Instead, it is flexible, selective, and responsive, allowing the cell to interact with its surroundings while preserving its own internal balance. At this early stage of our exploration, there is no need to focus on complex details or specialized terminology. Instead, it is enough to appreciate that life depends on boundaries. Without them, the organized processes of the cell would dissolve into randomness. Substances essential for survival would drift away, while harmful materials could enter freely. The simple presence of a boundary transforms a collection of molecules into a living system capable of persistence and adaptation. As scientists first peered through early microscopes, they encountered cells as small compartments, often noticing only their outlines. Over time, these outlines came to represent something far more meaningful than a simple edge. They marked the place where life defines itself, where order is preserved amid constant motion. The membrane surrounding the cell is not merely a wall. It is a living interface, a region of constant exchange and subtle control. In this calm session, we will gradually explore how this boundary functions, how it allows certain substances to pass while gently refusing others, and how it adapts to the needs of the cell over time. But for now, it is enough to rest with the idea that every cell exists in quiet conversation with its environment. Nothing inside the cell is truly isolated from what lies beyond, and yet nothing is allowed to move without purpose. Within the watery surroundings of the body, cells are bathed in fluids rich with dissolved nutrients, ions, and signaling molecules. These substances press softly against the outer surface of each cell, creating a constant potential for interaction. The cell membrane responds not with force, but with discernment. It allows water to flow where needed, permits nutrients to enter when required, and releases waste without disruption. This balance between openness and protection is one of the most elegant solutions life has ever produced. It may be soothing to consider that this balance is not maintained by conscious effort or intention. Cells do not think or decide in the way humans do. Instead, they follow physical and chemical principles that unfold naturally. Gradients form, molecules drift, proteins change, shape, and energy flows from one form to another. Through these simple processes, repeated endlessly, life sustains itself. As we remain at this introductory level, the emphasis is not on memorization or mastery, but on familiarity. The more gently we become acquainted with the cellular world, the less mysterious it seems. The cell is not an abstract concept or a distant scientific idea. It is the fundamental expression of life, present in every tissue, every organ, and every system of the body. In the quiet hours of rest, long after conscious activity has slowed, cells continue their work. They repair damage, regulate chemical levels, and preserve the conditions that allow the organism to awaken, renewed. This continuous activity depends, above all, on the cell's ability to manage what crosses its boundary. Without that ability, the rhythms of life would falter. As you listen, there is no need to hold each idea tightly. Let the concepts pass through your awareness as gently as molecules drifting through fluid. The details will return again and again in different forms, reinforcing themselves without urgency. Learning, in this context, is not about effort. It is about exposure, repetition, and calm attention. The microscopic world does not rush. Its processes unfold at their own pace, governed by natural laws that have remained unchanged for billions of years. Cells today function much as they did in the earliest forms of life, preserving internal conditions while responding to external change. This continuity links all living things across time, creating a quiet thread that runs through the history of life itself. As this session continues, we will move closer to the membrane itself, examining its structure, its components, and the mechanisms that allow substances to pass through it. For now, simply rest with the understanding that life depends on gentle boundaries, on selective openness, and on balance maintained moment by moment. In settling into this microscopic perspective, you may find that the mind itself begins to slow, mirroring the steady, unhurried processes of the cell. Just as the membrane filters and regulates without strain, your thoughts can soften, allowing only what is necessary to remain. The rest can drift away, leaving behind a quiet awareness, ready to continue this calm journey inward. 2. The cell as a self-contained system. As we continue gently forward, it becomes useful to imagine the cell not simply as a small object, but as a complete and self-sustaining system. Within its thin outer boundary, a cell carries everything it needs to survive, adapt, and persist, at least for a time. This self-contained nature is one of the defining characteristics of life. It allows the cell to exist as an individual unit, even while remaining deeply connected to the larger organism or environment around it. Inside the cell, countless processes unfold simultaneously. Molecules are built and broken down. Energy is captured and released. Signals are received and interpreted. These activities occur in a carefully organized manner, not because the cell plans them, but because the internal environment is kept within narrow limits. Temperature, acidity, ion concentration, and water balance are all regulated with remarkable consistency. This steady internal state allows chemical reactions to proceed reliably, day after day, moment after moment. Without regulation, the chemistry of life would quickly become unstable. Enzymes, the proteins that guide chemical reactions, are especially sensitive to their surroundings. Small changes in temperature or pH can alter their shape, reducing their effectiveness or stopping them altogether. For this reason, the cell must maintain conditions that support these delicate structures. The cell membrane plays a central role in this effort, acting as both a barrier and a mediator between the inside and the outside. The idea of the cell as a self-contained system does not imply isolation. Instead, it highlights controlled interaction. The cell must constantly exchange materials with its environment in order to survive. Nutrients must enter, waste must leave, and signals must be received. Yet this exchange is never random. Each movement across the membrane is governed by physical principles and molecular structures that ensure balance is preserved. This balance is often described using the term homeostasis, a concept that refers to the maintenance of stable internal conditions despite external change. At the level of the whole organism, homeostasis involves systems such as temperature regulation and blood glucose control. At the level of the cell, it involves ion gradients, water content, and molecular concentrations. The same principle applies at both scales, reflecting the deep continuity between cellular life and the larger structures it supports. To appreciate how remarkable this is, it helps to consider the environment in which most cells exist. In the human body, cells are surrounded by extracellular fluid, a constantly shifting mixture of water, dissolved salts, nutrients, and signaling molecules. The composition of this fluid can change with diet, activity, hydration, and health. Yet within the cell, conditions remain far more stable. This stability is not accidental. It is actively maintained through selective transport across the membrane. The membrane allows the cell to create internal conditions that differ from those outside. For example, certain ions may be far more concentrated inside the cell than outside, or vice versa. These differences are essential for processes such as nerve signaling, muscle contraction, and energy production. Maintaining them requires careful control, since diffusion naturally tends to eliminate concentration differences. The cell counters this tendency by using specialized proteins and energy-driven mechanisms. Even without delving into those details, it is calming to recognize that the cell's internal world is shaped by quiet, consistent effort. Nothing dramatic or forceful is required. Instead, the cell relies on countless small actions, each one modest on its own, yet powerful in combination. Molecules drift, channels open and close, and gradients are restored, all without interruption. The self-contained nature of the cell also allows for specialization. In multicellular organisms, different cells perform different roles, yet each one maintains its own internal balance. A nerve cell, a muscle cell, and a skin cell may look very different and perform distinct functions, but all rely on the same basic principles of membrane regulation. Their differences arise not from abandoning these principles, but from applying them in specific ways. This universality is part of what makes cell biology so soothing to study. Beneath the diversity of life lies a shared architecture, a common strategy for sustaining existence. Every cell, regardless of its origin, faces the same fundamental challenge: how to remain organized in a world that tends toward disorder. The solution, refined over billions of years, is the creation of a regulated internal environment, protected by a selective boundary. As we rest with this idea, it becomes easier to see the membrane not as a passive structure, but as an active participant in life. It senses changes, responds to signals, and adjusts its behavior accordingly. Yet it does so without awareness or intention, guided entirely by physical interactions and molecular design. This quiet efficiency is one of the most elegant features of living systems. The cell's ability to remain self-contained also allows it to endure temporary disruptions. Changes in the external environment do not immediately threaten survival because the membrane buffers the cell against sudden shifts. Only when these changes exceed the membrane's capacity to compensate does damage occur. This buffering capacity gives cells resilience, allowing them to function across a range of conditions. As you listen, you may notice a parallel between this cellular stability and the process of rest itself. Just as the cell maintains its internal environment amid external fluctuations, the mind can maintain a calm center even as thoughts arise and fade. Like molecules at the membrane, they can be allowed to pass gently without disturbing the deeper balance beneath. In time, we will explore the specific structures that make this regulation possible. We will look at the components of the membrane, the proteins that guide, transport, and the forces that drive movement across cellular boundaries. For now, it is enough to understand the purpose they serve. They allow the cell to exist as a coherent whole, capable of sustaining life through quiet, continuous regulation. The cell, as a self-contained system, is not a closed box, but a living balance point. It is open enough to interact with the world, yet closed enough to preserve its identity. This balance has been maintained across countless generations, linking the earliest cells to those that exist today. In recognizing this continuity, we begin to see life not as a collection of isolated events, but as a steady, unbroken process. As this idea settles, allow it to remain gently in the background. There is no need to hold on to every detail. The understanding will deepen naturally as we continue, returning to these principles again and again, each time from a slightly different angle. For now, simply rest with the image of the cell, quietly sustaining itself, moment by moment, through balance, regulation, and gentle exchange. 3. The purpose of the cell membrane. As our attention moves closer to the outer edge of the cell, it becomes clear that this boundary exists for a reason far deeper than simple separation. The cell membrane defines what it means to be a cell. It is the quiet structure that allows life to persist in a world governed by constant motion. Without it, the internal organization of the cell would dissolve, and the careful chemistry that sustains life would quickly fall apart. At its most basic level, the membrane establishes an inside and an outside. This distinction may seem simple, yet it is one of the most profound achievements of living systems. By creating a protected internal space, the cell gains the ability to regulate its own conditions. It can concentrate certain molecules, exclude others, and create gradients that drive essential processes. In doing so, the membrane transforms a collection of molecules into a living entity. The environment surrounding a cell is rarely stable. In the body, nutrient levels rise and fall, ion concentrations shift, and signaling molecules appear and disappear. Outside the body, conditions can be even more variable. Temperature, moisture, and chemical composition may change rapidly. The cell membrane allows life to persist despite this variability. It acts as a buffer, smoothing out sudden changes and allowing the cell time to respond. This buffering does not involve rigidity. The membrane is not a fixed wall resisting the outside world. Instead, it is flexible and responsive, constantly adjusting its behavior. It opens pathways when resources are needed and closes them when conditions become unfavorable. This adaptability is essential, as life depends on interaction just as much as it depends on protection. One of the membrane's most important purposes is regulation. The cell must maintain specific concentrations of ions such as sodium, potassium, and calcium. These ions play crucial roles in processes ranging from energy production to communication between cells. Left unchecked, they would simply diffuse until concentrations equalized across the membrane. The membrane prevents this outcome, allowing the cell to preserve the gradients that make life possible. Water balance is another quiet but essential function of the membrane. Water moves easily across many biological membranes, yet its movement must still be regulated. Too much water entering the cell can cause it to swell, while too little can lead to dehydration. The membrane ensures that water movement remains balanced, preserving the shape and integrity of the cell. Beyond regulation, the membrane also serves as a platform for communication. Cells do not exist in isolation, even when they function as independent units. They receive signals from their surroundings and from neighboring cells, allowing them to coordinate their activities. These signals often arrive at the membrane in the form of specific molecules that bind to proteins embedded in its surface. In this way, the membrane becomes a site where information is received and translated into action. This signaling function allows cells to respond appropriately to their environment. A muscle cell can contract when stimulated, a gland cell can release hormones, and an immune cell can recognize a potential threat. All of these responses begin at the membrane, where the external message first meets the internal machinery of the cell. The membrane also plays a role in identity. Cells must recognize which signals to respond to and which to ignore. They must distinguish between molecules meant for them and those intended for others. The membrane surface carries specific markers that help establish this identity. These markers allow cells to recognize one another, adhere to appropriate partners, and avoid unwanted interactions. In multicellular organisms, this recognition is especially important. Cells must cooperate to form tissues and organs, each performing its role within a larger system. The membrane enables this cooperation by providing a controlled interface for interaction. It ensures that cells remain distinct while still functioning as part of a whole. From a broader perspective, the membrane represents a solution to a universal challenge faced by all living systems. The laws of physics favor disorder, molecules naturally spread out, concentrations equalize, and organized structures break down over time. Life persists by continually opposing this tendency, maintaining order through constant effort. The membrane is central to this effort, allowing the cell to concentrate energy and matter where they are needed. It is comforting to realize that this effort does not require struggle or force. The membrane's functions arise from its structure and composition, from the way its components interact with water and other molecules. Physical principles guide its behavior, ensuring that regulation occurs naturally and efficiently. Life flows from these principles, rather than fighting against them. As we consider the purpose of the membrane, it becomes clear that it is both protective and enabling. It does not merely keep harmful substances out, it also allows beneficial ones in, supports communication, and preserves the delicate balance required for life. Protection without exchange would lead to stagnation, while exchange without protection would lead to chaos. The membrane achieves a quiet harmony between these extremes. This harmony has been refined over immense spans of time. The earliest cells likely possessed simple membranes, yet even these were sufficient to create internal environments distinct from the surrounding world. Over billions of years, membranes became more complex, incorporating proteins and other components that expanded their capabilities. Despite this complexity, the fundamental purpose of the membrane has remained unchanged. In reflecting on this, there is a sense of continuity that can be deeply calming. The same principles that govern the membranes of human cells also apply to those of bacteria, plants, and countless other forms of life. Across this diversity, the membrane stands as a shared solution, a common thread woven through the story of life. As you listen, there is no need to rush toward detailed mechanisms or technical explanations. Those will come in time, returning again and again in different forms. For now, it is enough to understand why the membrane exists at all. It allows the cell to be a cell, to sustain itself, and to participate in the larger patterns of life. The purpose of the cell membrane is not dramatic or grand. It is quiet and constant, unfolding moment by moment. It preserves balance, enables communication, and maintains identity, all without drawing attention to itself. In this way, it mirrors many of the processes that sustain life as a whole. As this understanding settles, you may find that your own thoughts begin to mirror the membrane's gentle selectivity. Some ideas pass through, others drift away, and a calm internal balance remains. There is nothing more to do in this moment than allow this awareness to rest, preparing gently for the next step in our exploration of the living cell. 4. Early ideas about cellular boundaries. As we continue gently onward, it is helpful to step back in time, to a period when the idea of a cell membrane was not yet clear or fully formed. The concept of a boundary surrounding every living cell did not arrive all at once. Instead, it emerged slowly, shaped by observation, experimentation, and gradual refinement. This slow unfolding mirrors the pace of biological discovery itself, steady rather than sudden, built on patience and quiet attention. When early scientists first examined living tissues through primitive microscopes, cells appeared as small compartments, often outlined by darker edges, but the nature of those edges remained mysterious. Some believed the boundary was little more than a passive skin, while others thought it might be an artifact of preparation rather than a true structure. The idea that this thin outline could play an active role in life was not immediately apparent. In the 17th century, when microscopes were still a novelty, cells were described as tiny rooms or chambers, enclosed spaces within plant and animal tissues. These early descriptions gave rise to the very word cell, borrowed from the Latin term for a small room. Yet the boundary itself was often overlooked, seen simply as the edge of the room rather than as something worthy of study in its own right. As microscopy improved, scientists began to notice that cells behaved differently from their surroundings. Certain dyes entered cells easily, while others were excluded. Some substances appeared to accumulate inside cells, while others remained outside. These observations hinted that the boundary was selective rather than open, capable of distinguishing between different molecules. Still, the mechanism behind this selectivity remained unclear. In the 19th century, advances in chemistry and physiology began to influence how scientists thought about cells. The cell was increasingly viewed as a chemical system, with reactions occurring in solution. This raised an important question. How could a chemical system maintain conditions different from those of its surroundings without some form of barrier? The idea of a semi-permeable boundary began to take shape, suggesting that the cell's outer edge allowed certain substances to pass while restricting others. Experiments with osmosis provided some of the earliest clues. When cells were placed in solutions of varying concentration, they swelled or shrank in predictable ways. These changes could not be explained without assuming the presence of a boundary that allowed water to move but limited the movement of other substances. The behavior of cells in these experiments pointed toward a membrane-like structure, even if its composition was still unknown. At the same time, studies of artificial membranes helped shape scientific thinking. Thin films made from natural materials could mimic some of the behaviors observed in cells, allowing water to pass while blocking larger molecules. These simple models suggested that the cell boundary might be more like a film or layer than a rigid wall. The idea that membranes were flexible and dynamic slowly began to replace older notions of solid enclosures. Despite these insights, early theories about the nature of the membrane varied widely. Some proposed that it was composed of proteins, others suggested lipids, and some believed it was a complex mixture of many substances. Without direct visualization, these ideas remained speculative. The membrane was inferred from behavior rather than seen directly, making it a concept grounded in interpretation as much as observation. As the 20th century approached, new experimental techniques brought greater clarity. Measurements of electrical properties revealed that cell boundaries could hold electrical charges and maintain voltage differences. This discovery was especially important for understanding nerve cells, which rely on electrical signals to communicate. The existence of a membrane capable of separating charges provided strong evidence that the boundary was not only real, but functionally essential. Chemical analysis further revealed that lipids played a major role in membrane structure. When lipids were extracted from cells and spread on water, they formed thin layers with properties similar to those expected of a cellular boundary. These findings suggested that the membrane might be composed, at least in part, of lipid molecules arranged in an orderly fashion. This was a crucial step toward understanding membrane structure. Still, the membrane was often imagined as a relatively simple layer, a passive barrier separating inside from outside. The idea that it might contain specialized structures for transport and communication had not yet fully emerged. Proteins were known to exist in cells, but their role in the membrane was not immediately recognized. It would take further experimentation and more refined models to reveal the membrane's true complexity. Throughout this period, the development of membrane theory reflected a broader shift in biology. Life was no longer seen as a collection of static parts, but as a dynamic system governed by physical and chemical principles. The membrane fit naturally into this view, acting as a site where these principles were applied to maintain organization and balance. What is striking about these early ideas is how much they relied on indirect evidence. Scientists could not see the membrane clearly, yet they inferred its existence from consistent patterns of behavior. This reliance on careful observation and logical reasoning is a reminder that scientific understanding often advances through patience rather than certainty. As you rest with this historical perspective, it may feel comforting to know that understanding does not require immediacy. Just as scientists slowly uncovered the nature of the cell membrane, learning can unfold gradually, without pressure or urgency. Each idea builds on the last, forming a coherent picture over time. The early history of membrane research also highlights the importance of asking simple questions. How does a cell keep its contents inside? Why do some substances enter while others do not? These questions, modest in appearance, open the door to an entire field of study. They remind us that curiosity, when paired with careful attention, can lead to profound insights. As the concept of the membrane became more established, it set the stage for deeper exploration. Researchers began to ask not only what the membrane was made of, but how it functioned, how it changed, and how it supported the life of the cell. These questions would eventually lead to detailed models and experimental techniques capable of revealing the membrane structure at the molecular level. For now, it is enough to appreciate the quiet progression of ideas that brought us here. The cell membrane, once an indistinct outline under a simple microscope, emerged as a central feature of life through decades of careful study. Its recognition was not the result of a single discovery, but of many small observations woven together. As this part draws to a close, allow the sense of gradual understanding to linger. There is no need to rush ahead. The membrane will reveal itself more fully as we continue, layer by layer, idea by idea. In this calm unfolding, both the history of science and the process of learning find a gentle rhythm, steady and unhurried, inviting rest as much as insight. 5. The fluid mosaic model. As understanding of cellular boundaries continued to deepen, scientists gradually realized that the membrane could not be a rigid or uniform structure. Its behavior suggested something far more dynamic, something capable of movement, flexibility, and change. This realization eventually led to what is known as the fluid mosaic model, a way of describing the membrane that captures both its stability and its quiet motion. The word fluid is used deliberately. It reflects the idea that the membrane behaves less like a solid wall and more like a gentle, flowing surface. Within this thin boundary, many of the components are free to move sideways, drifting slowly in response to their surroundings. This lateral movement allows the membrane to adapt, repair itself, and respond to changes without losing its overall integrity. The membrane holds together, yet it is never frozen in place. The term mosaic complements this idea of fluidity. Rather than being made of a single substance, the membrane is composed of many different components arranged together, much like pieces in a mosaic artwork. These pieces include lipids, proteins, and carbohydrates, each contributing to the membrane's function. No two regions of the membrane are exactly the same, and this variation allows the cell to perform many tasks at once. Before this model was proposed, membranes were often imagined as relatively simple layers, perhaps composed of proteins or lipids alone. However, such models could not fully explain the wide range of behaviors observed in living cells. The fluid mosaic model brought these observations together into a coherent picture, one that accounted for selective permeability, signaling, and flexibility all at once. At the heart of this model lies the idea that the membrane's basic structure is formed by a lipid bilayer. These lipids create a continuous foundation, a sort of calm sea in which other components float. Embedded within this sea are proteins of various shapes and sizes. Some span the membrane entirely, forming pathways across it, while others rest more loosely at the surface. Together, they create a pattern that is both organized and adaptable. What makes this arrangement especially effective is that it balances order with motion. The lipids are arranged in a predictable way, providing a stable barrier, yet they are not locked into place. They can shift position, allowing the membrane to bend, stretch, and even heal small disruptions. This fluid nature is essential for processes such as cell division, movement, and interaction with neighboring cells. The proteins within the membrane also benefit from this mobility. Because they are not fixed, they can cluster together, separate, or change orientation as needed. This flexibility allows cells to respond quickly to signals from their environment. When a signal arrives, the relevant proteins can interact efficiently, triggering internal responses without delay. It is soothing to consider how quietly this motion occurs. At the molecular level, there is constant activity, yet it unfolds smoothly, without noise or strain. Molecules drift, collide, and separate in patterns governed by simple physical forces. Over time, these countless small movements give rise to the complex behaviors we associate with life. The fluid mosaic model also helps explain how membranes maintain their selective nature. While the lipid bilayer forms a general barrier, proteins create specific pathways for certain substances. This arrangement allows the membrane to be both protective and interactive. Instead, their movement is guided by the presence or absence of appropriate proteins, ensuring that balance is preserved. Another important aspect of this model is its universality. While the specific composition of membranes can vary between cell types and organisms, the overall pattern remains the same. This consistency reflects the effectiveness of the design. Once evolution arrived at a structure capable of balancing stability and flexibility, it was refined rather than replaced. As scientific tools improved, evidence for the fluid mosaic model continued to grow. Techniques that allowed researchers to track the movement of membrane components showed that proteins and lipids indeed drifted laterally over time. These observations confirmed that the membrane was not static, but alive with motion, even in cells at rest. This constant motion does not lead to disorder. It is a reminder that order in living systems does not require stillness. In reflecting on this model, it can be helpful to draw a parallel to learning itself. It moves, shifts, and rearranges over time, gradually forming a coherent picture. Just as membrane components drift into useful arrangements, ideas settle into place through repeated exposure and quiet reflection. The fluid mosaic model also underscores the importance of context. A membrane's behavior depends on its environment, including temperature, chemical composition, and the presence of other molecules. Changes in these conditions can alter membrane fluidity, affecting how easily components move. Cells respond to these changes by adjusting the composition of their membranes, preserving function across varying circumstances. This adaptability is especially important in organisms that experience temperature changes. Membranes must remain fluid enough to function, even in cooler conditions, yet stable enough to maintain integrity when temperatures rise. The ability to fine-tune membrane composition allows cells to survive and thrive across a wide range of environments. As you rest with these ideas, there is no need to visualize every molecule or remember every detail. Instead, allow the overall impression to remain. The membrane is not a rigid barrier, nor is it a chaotic mixture. It is a calm, dynamic surface, shaped by physical principles and refined through time. The elegance of the fluid mosaic model lies in its simplicity. With a few core ideas, it explains a wide range of cellular behaviors. It shows how the membrane can be both strong and flexible, both selective and responsive. In doing so, it provides a foundation for understanding everything that follows. As this part gently concludes, take a moment to let the image of the membrane settle in your mind. Imagine a quiet surface, alive with slow motion, maintaining balance without effort. This image will return often as we continue, offering a stable reference point as we explore the many ways cells interact with their surroundings. 6. Phospholipids and bilayer formation. As our focus settles more closely on the structure of the cell membrane, we arrive at the molecules that quietly form its foundation. These molecules are called phospholipids, and though they are small, their collective behavior gives rise to one of the most important structures in all of biology. Without phospholipids, there would be no stable membrane, no clear separation between inside and outside, and no reliable way for life to maintain balance. A phospholipid is a simple molecule with two distinct regions, each with very different preferences. One end is attracted to water, while the other avoids it. This dual nature is not accidental. It is precisely what allows phospholipids to organize themselves so consistently and so effortlessly in watery environments. Wherever water is present, phospholipids respond in predictable ways, guided by basic physical forces rather than conscious direction. The water-attracting portion of the phospholipid is often described as a head. It interacts easily with surrounding water molecules, forming gentle associations that stabilize its position. The water-avoiding portion consists of two long tails made of fatty acids. Instead, they seek one another, clustering together in ways that minimize their exposure to the surrounding fluid. When phospholipids are placed in water, this contrast in behavior drives spontaneous organization. The heads turn outward toward the water, while the tails turn inward, away from it. When many phospholipids gather together, they arrange themselves into a double layer. In this structure, the water-loving heads face outward on both sides, while the water-avoiding tails meet in the center, forming a quiet, protected interior. This bilayer forms without instruction or energy input. It emerges naturally from the interaction between the molecules and their environment. This simplicity is part of its elegance. Life does not need to force membranes into existence. They assemble themselves wherever the conditions are right. Over and over again, across countless cells and organisms, the same structure appears, reliable and consistent. The phospholipid bilayer creates a barrier that is stable yet flexible. The tightly packed tails in the center resist the passage of many substances, especially those that are charged or strongly attracted to water. At the same time, the membrane remains thin and fluid, allowing for subtle movements and adjustments. This balance between resistance and flexibility is essential for cellular function. It is worth pausing to appreciate how different this structure is from rigid walls or solid shells. The membrane does not rely on hardness for strength. Instead, it relies on cooperation among many small components. Each phospholipid contributes to the whole, and the whole remains intact as long as the basic arrangement is preserved. Individual molecules can move, shift, or even be replaced without compromising the overall structure. Within the bilayer, phospholipids are not fixed in place. They drift slowly alongside one another, moving laterally within their layer. This movement allows the membrane to remain fluid, adapting its shape as the cell grows, divides, or changes position. The bilayer can bend without breaking, stretch without tearing, and reseal itself if briefly disrupted. This self-sealing property is especially important. Because phospholipids naturally reassemble into bilayers, these disruptions are often repaired automatically. The molecules simply rearrange themselves, restoring continuity without effort or awareness. The composition of phospholipid tails also influences membrane behavior. Some tails are straight, allowing them to pack closely together, while others are bent, creating more space between neighboring molecules. These differences affect how fluid the membrane is. Cells can adjust the types of phospholipids they use, fine-tuning membrane flexibility to suit their needs and environment. In warmer conditions, membranes tend to become more fluid, while in cooler conditions, they may stiffen. By altering phospholipid composition, cells maintain a comfortable balance, ensuring that membranes remain functional across a range of temperatures. This quiet adaptability supports life in environments that vary widely, from warm internal tissues to cooler external surroundings. The bilayer also establishes the basic permeability of the membrane. This selectivity provides a foundation for more complex transport mechanisms, which build upon the bilayer rather than replacing it. The membrane's ability to regulate movement begins here, with the simple arrangement of phospholipids. Although phospholipids form the core of the membrane, they do not act alone. Proteins and other molecules are embedded within or associated with the bilayer, adding layers of function. Yet even with these additions, the phospholipid bilayer remains central. It provides the stable surface upon which all other membrane activities depend. From an evolutionary perspective, this structure is remarkably ancient. Variations of phospholipid membranes appear in the earliest forms of life, suggesting that bilayer formation was one of the first solutions to the problem of cellular organization. Once established, it proved so effective that it has been conserved across billions of years. As you consider this, there is a quiet reassurance in knowing that life relies on such simple principles. The same forces that guide molecules in a glass of water also guide the formation of cellular boundaries. Complexity arises not from complication, but from repetition and refinement of basic patterns. In the context of rest and learning, this idea can feel grounding. There is no need for strain or effort to understand these concepts fully. Like phospholipids assembling into a bilayer, understanding forms naturally through gentle exposure. Each time the idea returns, it settles a little more firmly into place. The bilayer is not something the cell must constantly rebuild from scratch. Once formed, it persists, renewing itself gradually as individual molecules are replaced. This steady maintenance mirrors many processes in the body, where continuity is preserved through slow, ongoing renewal rather than sudden change. As this part draws to a close, allow the image of the phospholipid bilayer to remain softly in mind. Imagine countless small molecules quietly aligning themselves, heads outward, tails inward, forming a calm and reliable boundary. This boundary does not shout its presence, yet it supports every moment of cellular life. In the next stages of our journey, we will build upon this foundation, exploring how additional components interact with the bilayer to create the full range of membrane functions. For now, it is enough to rest with the simplicity and elegance of phospholipids, forming structure through balance, cooperation, and gentle motion. 7. Membrane fluidity and stability. As we remain with the image of the phospholipid bilayer, it becomes clear that structure alone is not enough to support life. A membrane must also possess the right balance between movement and firmness, between flexibility and persistence. This balance is known as membrane fluidity, and it is one of the quiet properties that allows cells to function smoothly over time. Without sufficient fluidity, membranes would become brittle and unresponsive. Without sufficient stability, they would lose their integrity. Life depends on holding these two qualities in gentle equilibrium. Membrane fluidity refers to the ease with which the molecules within the membrane can move. Phospholipids drift sideways within their layer, proteins shift position, and small regions of the membrane subtly rearrange themselves. This motion is not random chaos. It is constrained by molecular interactions and guided by physical principles. The result is a surface that is alive with quiet movement, yet reliably intact. This fluidity allows the membrane to adapt to changing conditions. When a cell changes shape, divides, or moves, its membrane must bend and stretch. A rigid structure would fracture under such demands. The fluid nature of the membrane ensures that these changes occur smoothly, without damage. Molecules slide past one another, redistributing themselves as needed, preserving continuity. Temperature plays a significant role in membrane fluidity. As temperature rises, molecular motion increases, and membranes tend to become more fluid. As temperature falls, movement slows, and membranes may become more rigid. Cells are sensitive to these changes, as membrane function depends on remaining within a certain range of fluidity. Too much fluidity can make the membrane leaky, while too little can hinder transport and signaling. To manage this, cells adjust the composition of their membranes. The fatty acid tails of phospholipids vary in length and shape. Some tails are straight, allowing them to pack closely together, while others contain bends that create space between neighboring molecules. Straight tails tend to reduce fluidity, while bent tails increase it. By altering the proportion of these different phospholipids, cells fine-tune membrane behavior. Another important contributor to membrane stability is a molecule called cholesterol, which is found in the membranes of many animal cells. Cholesterol fits between phospholipid tails, influencing how tightly they pack together. At higher temperatures, cholesterol helps restrain excessive movement, preventing the membrane from becoming too fluid. At lower temperatures, it disrupts tight packing, preventing the membrane from becoming too rigid. In this way, cholesterol acts as a quiet stabilizer, smoothing out extremes. The presence of cholesterol highlights an important theme in biology. Stability is often achieved not by rigidity, but by moderation. Rather than locking the membrane into a fixed state, cholesterol allows it to remain flexible across a range of conditions. This flexibility supports consistent function, even as the environment changes. Membrane proteins also contribute to both fluidity and stability. Some proteins anchor the membrane to internal structures within the cell, providing additional support. Others cluster together in specific regions, creating functional zones without immobilizing the entire surface. These interactions help organize the membrane while preserving its overall fluid nature. Despite this organization, membranes are never static. Even in cells at rest, molecules continue to move, exchange positions, and respond to subtle forces. This constant motion is essential for processes such as transport and signaling. When a signal arrives at the membrane, proteins may need to come together quickly. Fluidity allows this to happen efficiently, enabling rapid communication without disruption. Stability, on the other hand, ensures that the membrane remains a reliable boundary. The bilayer does not dissolve or fragment under normal conditions. Its overall structure persists, even as individual components come and go. This persistence is what allows cells to maintain their identity over time, replacing old molecules with new ones without losing coherence. The balance between fluidity and stability also affects how membranes interact with their surroundings. In some regions, membranes may be slightly more fluid, allowing for greater interaction with external molecules. In other regions, they may be more stable, providing protection or anchoring key structures. This subtle variation across the membrane adds another layer of functional complexity. From a broader perspective, membrane fluidity illustrates how life operates through gradients rather than absolutes. There is no single ideal state that applies at all times. Instead, cells continuously adjust, maintaining conditions within a range that supports function. This approach allows for resilience and adaptability, qualities that are essential for survival. It can be soothing to reflect on how quietly this adjustment occurs. Cells do not struggle to maintain balance. They simply respond to physical conditions, guided by molecular interactions that have been shaped through evolution. There is no urgency, no alarm, only steady regulation unfolding moment by moment. In the context of rest, this idea can feel reassuring. Just as the membrane finds balance without effort, the mind can also settle into a state of gentle equilibrium. Thoughts may move and shift, but a deeper stability remains beneath them. There is no need to force stillness. Calm emerges naturally when conditions are right. Membrane fluidity also plays a role in how cells age and respond to stress. Changes in membrane composition can affect sensitivity to damage, the efficiency of transport, and the responsiveness of signaling pathways. Maintaining appropriate fluidity helps protect cells over time, supporting long-term function. As we move forward in our exploration, We will see how this balance supports specific transport processes. Channels and carriers depend on the surrounding membrane environment to function properly. Too much rigidity or too much fluidity can disrupt their activity. The membrane provides not only a physical setting, but a carefully tuned context for these proteins to operate. For now, it is enough to appreciate the membrane as a living surface, one that is neither fixed nor fragile. It moves gently, holds together reliably, and adjusts continuously. This quiet adaptability underlies everything that follows, from the movement of ions to the transmission of signals. As this part gently concludes, allow the image of the membrane to remain soft and fluid in your mind. Imagine a surface that bends without breaking, shifts without losing form, and maintains balance without effort. In this image, there is no rush, no strain, only steady motion and quiet stability, supporting life one moment at a time. 8 membrane proteins and their roles. As our attention continues to rest on the living surface of the cell membrane, it becomes clear that phospholipids alone cannot account for all that the membrane accomplishes. While the bilayer provides structure and basic selectivity, it is the presence of proteins that gives the membrane much of its functional depth. These proteins are woven into the fluid surface, each one contributing quietly to the life of the cell. Membrane proteins are diverse in shape, size, and purpose, yet they share a common setting. They exist within or alongside the phospholipid bilayer, interacting both with the membrane itself and with the environment on either side. Some proteins extend completely across the membrane, while others associate more loosely with its surface. Together, they form a dynamic network that allows the cell to sense, respond, and regulate. One of the most important roles of membrane proteins is transport. Many substances essential for life cannot pass freely through the lipid bilayer. Charged particles, large molecules, and highly water-attracted substances require assistance. Membrane proteins provide this assistance, forming pathways that allow specific materials to cross the boundary in controlled ways. Without these proteins, the cell would be isolated, unable to acquire nutrients or release waste efficiently. Beyond transport, membrane proteins also act as receptors. These proteins bind to specific molecules outside the cell, such as hormones or signaling compounds. When binding occurs, the protein changes shape, triggering a response inside the cell. This process allows cells to communicate with their surroundings, coordinating activities across tissues and organs. The membrane thus becomes a listening surface, sensitive to subtle chemical messages. Other membrane proteins serve as anchors, connecting the membrane to internal structures within the cell or to neighboring cells. These connections provide mechanical stability. They also allow cells to form organized groups, creating layers, networks, and complex structures that support higher-level functions. The presence of proteins within the membrane adds to its mosaic-like character. Each protein occupies a specific region, contributing to the overall pattern without disrupting the bilayer's integrity. Because the membrane is fluid, proteins can move laterally, clustering when needed, or dispersing when their task is complete. This mobility supports adaptability, allowing the cell to reorganize its surface in response to changing conditions. It is worth noting that membrane proteins do not insert themselves randomly. Their structure determines how they interact with the lipid environment. Portions of the protein that are comfortable among lipid tails embed themselves within the bilayer, while regions that interact with water extend outward. This arrangement allows proteins to integrate seamlessly into the membrane, becoming part of its functional landscape. The variety of membrane proteins reflects the complexity of cellular life. Some proteins specialize in transporting a single type of molecule, while others participate in broader regulatory processes. Some respond quickly to changes, while others maintain steady conditions over long periods. Together, they form a coordinated system that supports both immediate needs and long-term stability. From an evolutionary perspective, the integration of proteins into membranes represented a major advance. Early membranes composed primarily of lipids could provide basic separation, but the addition of proteins greatly expanded functional possibilities. Cells gained the ability to control transport with precision, communicate over distances, and respond dynamically to their environment. These capabilities laid the groundwork for the diversity of life we see today. Despite their importance, membrane proteins operate quietly. They do not announce their activity or demand attention. They simply perform their roles, moment after moment, guided by molecular interactions and physical principles. This quiet reliability is a recurring theme in cellular biology, where life is sustained not by dramatic events, but by continuous, subtle processes. As we rest with this idea, it can be comforting to know that complexity does not require chaos. The membrane is busy, yet orderly. Many different proteins work simultaneously without interfering with one another, each occupying its place within the fluid mosaic. This cooperation emerges naturally from structure and interaction, rather than from central control. Membrane proteins are also subject to renewal. This turnover allows the cell to adapt over time, adjusting the composition of its membrane to suit changing needs. Old or damaged proteins are removed, and new ones take their place, preserving function without disrupting continuity. This gradual renewal mirrors the broader patterns of life. Stability is maintained not by permanence, but by ongoing replacement and repair. The membrane remains functional even as its individual components change, illustrating how continuity can emerge from constant motion. In the context of rest and learning, this idea can feel reassuring. Understanding does not depend on holding on to every detail indefinitely. Instead, it builds through repetition and renewal. Ideas are revisited, refined, and integrated over time, much like proteins within the membrane. As we move forward, we will explore specific types of membrane proteins in more detail, focusing on how they guide substances across the membrane. For now, it is enough to appreciate their presence and purpose. They transform the membrane from a simple barrier into an active interface, capable of regulation. The membrane, enriched by proteins, becomes a site of quiet intelligence, not intelligence in the conscious sense, but in the sense of responsiveness and adaptability. It responds appropriately to signals, maintains balance, and supports the life of the cell without awareness or intention. As this part gently comes to rest, allow the image of the membrane to expand slightly. Imagine not just a bilayer of lipids, but a living surface dotted with proteins, each one performing its role with steady precision. There is no urgency, no strain, only continuous function unfolding calmly. In this image, the membrane becomes not just a boundary, but a bridge between the cell and the world around it, supporting life through balance, interaction, and quiet cooperation. 9 channels and carrier proteins. As we continue our slow exploration across the surface of the cell membrane, our attention naturally settles on 2 groups of membrane proteins that play especially important roles in transport. These are channels and carrier proteins. Together, they provide the cell with controlled pathways, allowing substances to cross the membrane in an orderly and balanced way. Without them, the membrane would be largely closed to many of the materials essential for life. The phospholipid bilayer forms an effective barrier, particularly against charged particles and large molecules. While this barrier is vital for maintaining internal conditions, it also presents a challenge. Cells require a steady exchange of ions, nutrients, and other substances with their environment. Channels and carriers resolve this challenge by offering selective routes through the membrane, ensuring that exchange occurs without compromising stability. Channel proteins create narrow, water-filled passages that extend across the membrane. These passages allow specific substances to move from one side to the other, guided primarily by concentration differences. The interior of a channel is shaped and charged in a way that favors certain molecules while excluding others. This selectivity ensures that only appropriate substances are permitted to pass. Many channels are designed for ions such as sodium, potassium, calcium, or chloride. These ions are small, yet their electrical charge prevents them from slipping through the lipid interior of the membrane. Channels provide a safe route, shielding the ions from the surrounding lipids as they move across. The movement is smooth and efficient, requiring no direct input of energy from the cell. Some channels are always open, allowing continuous flow, while others are gated. Gated channels open and close in response to specific signals. These signals may include changes in voltage across the membrane, the binding of a particular molecule, or mechanical forces such as stretching. Through this gating behavior, cells gain precise control over when and where substances move. Carrier proteins operate in a different, more deliberate manner. Rather than forming open passages, carriers bind to specific molecules on one side of the membrane. This binding causes the protein to change shape, carrying the molecule across and releasing it on the other side. Once released, the protein returns to its original shape, ready to repeat the process. This cycle of binding, change, and release unfolds steadily, one molecule at a time. While slower than movement through open channels, carrier-mediated transport offers high specificity. Each carrier typically recognizes only one or a few closely related substances. This specificity helps the cell regulate its internal composition with remarkable precision. Carrier proteins are especially important for transporting larger molecules such as sugars and amino acids. These substances are vital for energy production and growth, yet they cannot pass through channels easily. Carriers provide a gentle, reliable solution, ensuring that these molecules reach the cell interior when needed. Both channels and carriers rely on gradients, differences in concentration or electrical charge, to guide movement. When a substance is more concentrated on one side of the membrane than the other, they do not force substances uphill. Instead, they provide a path of least resistance. This reliance on gradients reflects a broader theme in cellular life. Cells work with physical principles rather than against them. By harnessing natural tendencies, they achieve regulation without unnecessary effort. Energy is conserved, and balance is maintained through cooperation with the environment. The presence of channels and carriers also allows cells to respond rapidly to changing conditions. When a signal arrives or a need arises, transport proteins can increase or decrease their activity. Channels may open or close, and carriers may be produced or removed from the membrane. Through these adjustments, the cell fine-tunes its interactions with the outside world. It is important to note that transport through channels and carriers does not occur in isolation. Many proteins work together, creating coordinated responses. For example, the opening of an ion channel may trigger a cascade of events inside the cell, leading to changes in metabolism or gene expression. In this way, simple movements across the membrane can have far-reaching effects. Despite their importance, channels and carriers operate quietly. There is no accumulation of noise or disturbance as molecules pass through. Each movement is small, localized, and transient, yet the combined effect of countless such movements sustains the life of the cell. From a structural perspective, channels and carriers are shaped by the same principles that govern all proteins. The arrangement of their amino acids creates binding sites, pathways, and flexible regions that allow movement. Changes in shape are not random but precisely suited to the tasks these proteins perform. Over time, these proteins are replaced and renewed, just like other components of the membrane. This turnover allows cells to adapt, altering the types and numbers of transport proteins to match changing needs. A cell that requires more glucose, for example, may produce more glucose carriers, increasing uptake without altering the membrane's basic structure. As we consider this process, it becomes easier to appreciate the membrane as an active, responsive surface. It is not merely a backdrop for transport, but a participant in regulation. Channels and carriers are part of a larger system that maintains balance through constant, gentle adjustment. In the context of rest and learning, this idea carries a quiet reassurance. Understanding does not require force. Like molecules following gradients, comprehension emerges naturally when pathways are available. With repeated exposure, ideas pass into awareness, settle, and integrate without strain. As this part draws to a close, allow the image of channels and carriers to remain softly present. Imagine narrow pathways opening and closing, carriers gently shifting shape, and molecules moving calmly across the membrane. There is no rush, no excess, only steady exchange guided by balance. In the next stage of our exploration, we will look more closely at how this movement supports the larger principle of selective permeability. For now, it is enough to rest with the knowledge that channels and carrier proteins provide the cell with quiet, reliable routes of connection, sustaining life through precision, restraint, and gentle flow. 10 Carbohydrates and cellular recognition. As we continue drifting across the surface of the cell membrane, another layer of quiet complexity comes into view. Alongside lipids and proteins, there are carbohydrates, delicate chains of sugars that extend outward from the membrane into the surrounding environment. These carbohydrate structures may appear subtle, yet they play a vital role in how cells recognize one another, communicate, and maintain their place within the living system. Unlike lipids and proteins, membrane carbohydrates are almost always found attached to other molecules. They are linked either to proteins, forming structures known as glycoproteins, or to lipids, creating glycolipids. In both cases, the carbohydrate portion faces outward, projecting into the extracellular space like soft, branching identifiers. This outward orientation is not accidental. It positions carbohydrates perfectly for interaction with the external environment. These carbohydrate chains vary in length and structure, and this variation carries meaning. Each cell type displays a unique pattern of carbohydrates on its surface, a kind of molecular signature. This signature allows cells to distinguish between self and non-self, between familiar neighbors and foreign intruders. In this way, carbohydrates contribute to cellular identity in a quiet yet powerful manner. In multicellular organisms, this recognition is essential. Cells must adhere to the correct partners to form tissues and organs. Muscle cells, nerve cells, and epithelial cells must all find their appropriate places and maintain proper connections. Carbohydrate patterns help guide this organization, ensuring that cells assemble into functional structures rather than chaotic collections. The immune system relies heavily on carbohydrate recognition. Immune cells scan the surfaces of other cells, reading their carbohydrate patterns to determine whether they belong to the body or represent a potential threat. Pathogens often carry unfamiliar carbohydrate signatures, allowing immune cells to identify and respond to them. This recognition occurs without awareness or intent, guided entirely by molecular interactions. Carbohydrates also play a role in cell-to-cell communication. When cells send signals to one another, those signals often involve binding events at the membrane surface. Carbohydrate chains can influence these interactions, enhancing or limiting access to underlying proteins. In this way, carbohydrates help regulate how signals are received and interpreted. It is worth noting that carbohydrate chains are highly diverse. Even small differences in structure can have significant effects on recognition and binding. This diversity allows for fine distinctions between cell types and states. A cell undergoing development or responding to stress may alter its carbohydrate display, subtly changing how it interacts with its environment. Despite their importance, membrane carbohydrates are not rigid markers. Like other membrane components, they exist within a fluid context. The molecules they are attached to can move laterally within the membrane, allowing carbohydrate patterns to shift and reorganize. This mobility supports adaptability while preserving overall identity. From a structural perspective, carbohydrates extend into the watery environment outside the cell, forming a soft, protective layer, sometimes referred to as the glycocalyx. This layer cushions the cell, protecting it from mechanical stress and chemical damage. It also helps regulate interactions with surrounding molecules, creating a controlled interface between the cell and its environment. The glycocalyx can influence how cells interact with fluids, how they adhere to surfaces, and how they respond to physical forces. In blood vessels, for example, carbohydrate-rich surfaces help regulate the flow of blood cells, reducing friction and preventing unwanted adhesion. These effects occur continuously, without interruption, supporting the smooth operation of the body. Carbohydrates also play a role in development. As cells divide and differentiate, changes in carbohydrate expression help guide their paths. These changes influence how cells migrate, where they settle, and how they interact with neighboring cells. Through these quiet adjustments, complex structures emerge over time. It is calming to reflect on how much of this coordination occurs without central control. No single cell directs the process. Instead, patterns emerge from local interactions, guided by molecular compatibility. Carbohydrates contribute to this process by providing a language of recognition, a way for cells to identify appropriate partners and responses. From an evolutionary perspective, carbohydrate diversity has been shaped by countless interactions between organisms and their environments. Pathogens evolve to mimic host carbohydrates, hosts evolve new patterns, and the cycle continues. This ongoing exchange has refined the recognition systems that protect and organize life today. As we rest with these ideas, there is no need to memorize specific structures or pathways. The essential point is that carbohydrates add a layer of information to the membrane. They transform the surface of the cell into a communicative landscape, rich with meaning yet gentle in its operation. In the context of rest and learning, this idea can feel reassuring. Communication does not require force or noise. It can occur through subtle patterns, quiet signals, and gentle recognition. Just as cells recognize one another through carbohydrates, understanding can emerge through repeated, soft exposure rather than intense focus. As this part gently draws to a close, imagine the surface of the cell once more. Picture lipids forming a calm foundation, proteins moving and working quietly, and carbohydrates extending outward like delicate branches. Together, they create a living surface that protects, communicates, and identifies without effort or urgency. In the next stage of our journey, we will bring these components together to explore the principle of selective permeability. For now, allow the image of carbohydrate-rich membranes to settle softly, a reminder that even the smallest details contribute to the harmony and balance that sustain life. 11. Selective permeability. As the various components of the cell membrane come together in the mind, a unifying principle begins to emerge. This principle is known as selective permeability, and it captures the membrane's essential role in regulating exchange. The membrane does not allow everything to pass freely, nor does it block all movement. Instead, it permits some substances to cross with ease, restricts others, and carefully controls many more. Through this selectivity, the cell maintains balance, while remaining connected to its surroundings. Selective permeability is not the result of conscious choice. It arises naturally from the structure of the membrane and the properties of its components. The phospholipid bilayer forms a foundation that favors certain types of molecules over others. Small, nonpolar substances can often pass through the lipid interior with relative ease, while charged or large molecules are slowed or stopped. This basic filtering effect sets the stage for more precise control. Proteins embedded within the membrane refine this selectivity further. Channels and carriers create specific pathways for substances that would otherwise be excluded. Each pathway has its own preferences, shaped by the structure of the protein and the chemical nature of the substance it transports. In this way, the membrane becomes not just a barrier, but a gatekeeper, guiding traffic according to the needs of the cell. Water occupies a special place in this system. As the most abundant molecule in living systems, its movement must be carefully regulated. Water can cross many membranes relatively easily, yet its flow is still subject to control. The membrane's selective properties ensure that water movement supports balance rather than disrupting it. Too much or too little water can be harmful, and the membrane helps prevent extremes. Ions present another important case. These charged particles are essential for processes such as electrical signaling and energy production, yet their charge prevents them from passing through the lipid bilayer unaided. Selective permeability ensures that ions cross the membrane only through appropriate channels, preserving concentration gradients that are vital for cellular function. Nutrients such as sugars and amino acids are also subject to selective control. These molecules provide energy and building materials, yet they cannot simply diffuse into the cell. Carrier proteins recognize them specifically, transporting them across when conditions are right. This selectivity prevents unwanted substances from entering while ensuring that essential resources are available. Waste products, too, must be managed. Metabolic processes generate byproducts that can become harmful if allowed to accumulate. Selective permeability allows these wastes to exit the cell efficiently, maintaining internal cleanliness without compromising integrity. The membrane thus supports both intake and release, balancing the flow in both directions. What makes selective permeability especially elegant is its adaptability. Cells can alter the number and type of transport proteins they express, adjusting permeability in response to internal and external conditions. A cell that requires more nutrients may increase uptake pathways, while one under stress may restrict certain movements to protect itself. This adaptability allows cells to survive in changing environments. When external conditions shift, the membrane responds, modifying its selectivity to preserve internal stability. This response does not require foresight or planning. It unfolds naturally, guided by molecular interactions and regulatory processes that have been shaped through evolution. Selective permeability also supports communication. By controlling which signaling molecules can interact with receptors, the membrane influences how messages are received and processed. Some signals are allowed to pass or bind easily, while others are ignored. This filtering ensures that cells respond appropriately to relevant information without being overwhelmed by noise. In multicellular organisms, selective permeability contributes to coordination across tissues and systems. Different cell types display different permeability patterns, reflecting their specialized roles. A nerve cell's membrane behaves differently from that of a muscle cell or a gland cell, yet all rely on the same underlying principles. This diversity within unity is a hallmark of biological organization. From a physical perspective, selective permeability represents a balance between openness and protection. Absolute openness would lead to loss of internal organization, while complete closure would isolate the cell from resources and signals. The membrane occupies a middle ground, allowing exchange without surrendering control. This balance is maintained continuously, moment by moment. It is calming to reflect on how quietly this regulation occurs. There are no alarms or sudden shifts, only steady adjustments guided by gradients, affinities, and structural compatibility. The membrane does not fight the environment. It works with it, channeling natural tendencies into orderly outcomes. In the context of rest and learning, this principle can feel reassuring. The mind, like the cell, does not need to absorb everything at once. It can be selective, allowing useful ideas to enter while letting others pass by without disruption. Understanding builds through gentle filtering, not force. Selective permeability also highlights the importance of boundaries. Boundaries are not obstacles to connection, but conditions for meaningful interaction. By defining what crosses and what remains outside, the membrane creates the space in which life can unfold. Without boundaries, There would be no distinct internal world, no continuity, no sustained activity. As this idea settles, it may become easier to see boundaries in a new light. They are not signs of separation or exclusion, but tools for balance. They allow systems to remain open without becoming overwhelmed, to engage without losing identity. This principle applies not only to cells, but to many aspects of living systems. As we move forward, we will explore specific forms of transport that operate within this framework, beginning with processes that require no energy input. For now, it is enough to rest with the concept of selective permeability as a guiding theme, one that brings together structure, function, and balance. Imagine the membrane once more, gently regulating the flow of substances, opening and closing pathways as needed. There is no hurry, no excess. Everything moves according to quiet rules, preserving harmony within constant change. In this image, selective permeability becomes not just a scientific concept, but a reflection of life's calm and continuous rhythm. 12. As we continue gently forward, the idea of selective permeability begins to take on a more tangible form through the concept of passive transport. Passive transport describes the movement of substances across the cell membrane without the direct use of cellular energy. It is movement guided not by force, but by natural tendencies that arise whenever molecules are free to drift. In this way, passive transport reflects one of the most calming principles in biology. Balance emerges when systems are allowed to follow simple physical laws. At the molecular level, particles are never still. Even in seemingly quiet environments, molecules are in constant motion, colliding, separating, and redistributing themselves. This movement, driven by thermal energy, creates patterns that favor equilibrium. When a substance is more concentrated in one area than another, its molecules tend to spread out, moving from regions of higher concentration toward regions of lower concentration. Passive transport allows the cell to take advantage of this tendency rather than resisting it. The membrane plays a supportive role in this process. It does not push molecules across or pull them inward. Instead, it provides pathways or permits passage where appropriate, allowing natural movement to proceed. This cooperation between structure and physics allows the cell to exchange materials efficiently while conserving energy for tasks that truly require it. One of the most familiar examples of passive transport is diffusion. Diffusion occurs whenever molecules move down a concentration gradient, gradually spreading until they are evenly distributed. In the context of the cell membrane, diffusion allows small, non-polar molecules such as oxygen and carbon dioxide to pass directly through the lipid bilayer. These molecules dissolve easily in the membrane's interior, slipping quietly from one side to the other. This movement is continuous and gentle. Oxygen enters the cell as it is used for metabolism, while carbon dioxide leaves as a waste product. There is no need for gates to open or energy to be spent. The membrane simply allows these molecules to pass, maintaining balance through constant exchange. Other substances require assistance to move passively across the membrane. Charged particles and larger polar molecules cannot cross the lipid bilayer on their own, yet they may still move without energy input. In these cases, channel or carrier proteins provide a pathway, allowing diffusion to occur through a controlled route. This process is often referred to as facilitated diffusion. Though the movement itself remains passive, facilitated diffusion still depends on gradients. Molecules move in the direction favored by concentration or electrical differences. The proteins involved do not alter this direction. They simply reduce resistance, making movement possible without disrupting the membrane's barrier function. This quiet facilitation exemplifies how cells enhance efficiency without unnecessary effort. Water movement also falls under the umbrella of passive transport. While water can pass directly through the bilayer to some extent, specialized channels often increase the efficiency of this movement. Even so, The driving force remains the same. Water moves naturally toward balance. What makes passive transport especially elegant is its predictability. It simply allows movement to occur where appropriate. This predictability contributes to the stability of internal conditions, supporting consistent function over time. Passive transport also illustrates the importance of gradients in biological systems. Gradients represent stored potential, differences that can be used to drive processes without energy expenditure. By maintaining gradients, cells create opportunities for passive movement that support metabolism, signaling, and homeostasis. It may seem counterintuitive that maintaining gradients often requires energy, while using them does not. Yet, this balance is central to cellular economy. Energy is invested to establish and preserve gradients, and passive transport then takes advantage of them. This division of labor allows cells to operate efficiently, conserving resources while sustaining life. In the context of rest and learning, passive transport offers a gentle metaphor. There is no need to force comprehension. When pathways are available, insight drifts into place, guided by curiosity rather than effort. Passive transport also contributes to the quiet resilience of cells. Even when energy levels are low, passive processes can continue, supporting basic survival. This reliability adds a layer of security, ensuring that essential exchanges do not cease abruptly. The membrane's role in passive transport is one of subtle facilitation. It shapes the environment in which movement occurs, guiding substances along appropriate paths. In doing so, it maintains the delicate balance between openness and control that defines cellular life. As molecules move across the membrane through passive transport, they do so individually, each following its own path. Yet the collective effect of countless such movements shapes the cell's internal environment. This accumulation of small, simple events gives rise to stability and function on a larger scale. It is soothing to reflect on how little effort this requires. Passive transport does not strain or exhaust the cell. It unfolds quietly, moment by moment, guided by gradients that are already present. In this way, the cell aligns itself with the natural flow of energy, rather than opposing it. As this part gently settles, allow the idea of natural movement to remain in awareness. Imagine molecules drifting across the membrane, guided by gentle differences, moving steadily toward balance. There is no urgency, no resistance, only calm redistribution. In the next part of our journey, we will explore diffusion and facilitated diffusion in greater detail, examining how these processes support life across many contexts. For now, it is enough to rest with the understanding that passive transport represents one of the cell's most peaceful strategies, allowing life to continue through cooperation with the quiet laws of nature. 13. Diffusion and facilitated diffusion. As we continue calmly onward, the idea of passive transport becomes more defined through two closely related processes: diffusion and facilitated diffusion. These forms of movement are among the most fundamental activities taking place in every living cell. They occur continuously, quietly shaping the internal environment without drawing attention to themselves. Through them, cells maintain balance, exchange materials, and remain responsive to their surroundings, all while conserving energy. Diffusion is the simplest expression of molecular movement. It arises naturally from the constant motion of particles. Molecules move not with intention, but as a consequence of their kinetic energy, spreading from regions where they are more concentrated into regions where they are less so. Over time, this movement leads toward equilibrium, a state in which molecules are evenly distributed. In living systems, diffusion is never static, but ongoing. Within the context of the cell membrane, diffusion allows certain substances to cross directly through the phospholipid bilayer. Small, nonpolar molecules are particularly well suited to this process. Oxygen, for example, dissolves easily within the lipid interior of the membrane. As cells consume oxygen for metabolism, its internal concentration drops, creating a gentle gradient. Oxygen molecules outside the cell drift inward, restoring balance without any effort on the cell's part. Carbon dioxide behaves in a similar way, but in the opposite direction. As a waste product of metabolism, it accumulates inside the cell. Diffusion allows it to drift outward, leaving the cell and entering the surrounding fluid. This exchange occurs continuously, quietly supporting respiration at the cellular level. There are no gates to open, no signals to send. The membrane simply permits movement where chemistry allows. Not all substances are able to diffuse directly through the lipid bilayer. Many molecules essential for life are either too large or too strongly attracted to water to pass through the membrane's hydrophobic interior. Yet these substances may still move passively, provided the membrane offers assistance. This is where facilitated diffusion comes into play. Facilitated diffusion relies on membrane proteins to provide pathways across the membrane. These proteins do not change the direction of movement or add energy to the process. Instead, they lower the barrier that would otherwise prevent diffusion. By creating a suitable environment for the molecule to pass through, they allow natural movement to proceed. Channel proteins are one means by which facilitated diffusion occurs. These channels form pores that span the membrane, lined with amino acids that interact favorably with specific substances. Ions, for example, can move through channels that shield their charge from the surrounding lipids. As long as a gradient exists, ions flow through these channels smoothly, restoring balance. Carrier proteins provide another route for facilitated diffusion. Unlike channels, carriers bind to specific molecules and undergo a subtle change in shape. This change carries the molecule across the membrane, releasing it on the other side. The protein then returns to its original shape, ready to repeat the process. Each cycle unfolds gently, guided by the presence of the gradient. Both channels and carriers are selective. They do not allow all substances to pass, but only those that match their structural features. This selectivity ensures that facilitated diffusion remains controlled, supporting balance rather than undermining it. Even though movement is passive, it is not indiscriminate. One of the defining features of both diffusion and facilitated diffusion is that they proceed until equilibrium is reached. Once concentrations are equal on both sides of the membrane, net movement stops. Molecules continue to move back and forth, but there is no overall change. This behavior reflects the tendency of physical systems to settle into balanced states when left undisturbed. In living cells, true equilibrium is rarely achieved for long. Metabolic activity constantly alters concentrations, creating new gradients. As a result, diffusion and facilitated diffusion continue indefinitely, responding to these shifts. This dynamic balance supports life, allowing cells to adapt continuously without dramatic change. It is worth noting that facilitated diffusion can reach a limit. Because it relies on specific proteins, there are only so many pathways available at a given time. When all carriers or channels are occupied, the rate of transport cannot increase further, even if the gradient becomes steeper. This saturation reflects the physical presence of the proteins themselves. Simple diffusion through the lipid bilayer does not have this same limitation, but it is restricted by the properties of the membrane and the molecule involved. Together, these two forms of movement complement one another, providing both broad and specific routes for passive transport. From a broader perspective, diffusion and facilitated diffusion exemplify the cell's preference for efficiency. Whenever possible, the cell allows natural forces to do the work. Energy is reserved for processes that truly require it, such as moving substances against gradients or performing complex synthesis. This strategy supports sustainability at the cellular level. There is something deeply calming about this approach. Life does not constantly struggle against its environment. Instead, it flows with it, using gradients and movement to maintain order. Diffusion and facilitated diffusion are expressions of this philosophy, quiet processes that achieve much with little effort. In the context of rest and learning, these ideas can be gently reassuring. Understanding, like diffusion, spreads naturally when conditions are right. When the mind is receptive, ideas move in, settle, and balance themselves over time. There is no need to push or force comprehension. As we consider diffusion more closely, it becomes clear that it is not merely a laboratory concept, but a process that underlies countless physiological functions. Gas exchange in the lungs, nutrient absorption in tissues, and waste removal all rely, at least in part, on diffusion and facilitated diffusion at the cellular level. These processes also contribute to the remarkable coordination seen in multicellular organisms. By maintaining gradients across membranes, cells create conditions that support larger-scale functions, such as nerve signaling and muscle contraction. Each small movement contributes to a larger pattern, linking the microscopic to the macroscopic. As this part gently comes to rest, allow the idea of diffusion to remain softly present. Imagine molecules drifting, spreading, and settling, guided by gentle differences. Picture channels opening quietly, carriers shifting shape, and balance being restored without effort. In the next stage of our exploration, we will turn our attention to the movement of water and the concept of osmosis, another form of passive transport that plays a central role in cellular balance. For now, it is enough to rest with diffusion and facilitated diffusion, processes that sustain life through simplicity, cooperation, and calm, continuous motion. 14. Osmosis and water balance. As our attention continues to move gently across the processes of passive transport, we arrive at a form of movement so quiet and constant that it often goes unnoticed. This is osmosis, the movement of water across a selectively permeable membrane. Though simple in principle, osmosis plays a central role in maintaining cellular balance, shaping the internal environment in ways that support every other process of life. Water is the most abundant substance in living systems. It fills cells, surrounds them, and serves as the medium in which nearly all biological reactions occur. Because of this, the movement of water must be carefully regulated. Too much water entering a cell can cause it to swell, while too little can lead to shrinkage and stress. Osmosis is the process by which water naturally moves in response to differences in solute concentration, gently guiding itself toward balance. At its core, osmosis is a form of diffusion. Water molecules move from regions where they are more abundant to regions where they are less so. However, rather than focusing on water concentration alone, osmosis is best understood in terms of solutes, the dissolved substances within water. When solute concentration differs across a membrane, imagine two solutions separated by a membrane that allows water to pass but restricts many solutes. On one side, the solution contains fewer dissolved particles. On the other, it contains more. Water molecules, free to move, drift toward the side with higher solute concentration. In doing so, they dilute that solution, reducing the difference between the two sides. This movement continues until balance is reached or until other forces intervene. The cell membrane provides the perfect setting for osmosis. Its selective permeability allows water to move relatively freely while controlling the passage of solutes. Specialized proteins often increase the efficiency of water movement, but the driving force remains the same. Water follows gradients, responding quietly to differences in solute concentration. This movement of water has profound effects on cell shape and volume. When water enters a cell, the cell expands. When water leaves, the cell contracts. These changes are not merely cosmetic. They influence internal pressure, molecular interactions, and overall function. The membrane's ability to withstand and respond to these changes is a testament to its flexibility and strength. Cells exist in environments that can vary in solute concentration. In some cases, the surrounding fluid has a similar concentration to the cell's interior. In others, it may be more or less concentrated. Osmosis allows cells to respond to these conditions naturally, adjusting water movement to preserve balance. It is important to recognize that osmosis does not require energy input from the cell. Like diffusion, it unfolds according to physical principles. Water molecules move continuously, colliding, separating, and redistributing themselves. The membrane does not command this movement. It simply allows it, shaping the outcome through selective permeability. In living organisms, osmosis supports countless functions. It influences nutrient absorption, waste removal, and the maintenance of blood volume. At the cellular level, it ensures that enzymes and other molecules remain in appropriate concentrations, preserving the conditions needed for chemical reactions to proceed smoothly. Plant cells offer a particularly clear illustration of osmotic balance. Their rigid cell walls allow them to tolerate significant water influx without bursting. As water enters, internal pressure builds, supporting the plant's structure and keeping tissues firm. In animal cells, which lack such walls, osmotic balance must be more tightly regulated to prevent damage. Despite these differences, the underlying principle remains the same. Water moves in response to solute concentration, and the membrane guides this movement to maintain stability. This quiet regulation unfolds continuously, without interruption, sustaining life at every moment. Osmosis also highlights the interconnectedness of cellular processes. Changes in solute concentration often result from metabolic activity or transport of other substances. As solutes move, water follows, linking different forms of transport into a unified system. The cell does not manage each process in isolation. Instead, balance emerges from their interaction. There is something deeply calming about this image of water movement. It is steady, predictable, and gentle. Water does not rush or hesitate. It flows according to gradients, responding to conditions without resistance. In this way, osmosis embodies a kind of natural harmony, a tendency toward balance that supports life without effort. In the context of rest and learning, osmosis offers a soothing metaphor. There is no need to force ideas into place. With gentle exposure, they find their equilibrium, integrating quietly into awareness. Osmosis also reminds us that boundaries are essential for meaningful flow. Without a selectively permeable membrane, water movement would be uncontrolled, leading to disorder rather than balance. The membrane provides the structure within which natural movement can be beneficial rather than disruptive. As we rest with this idea, it becomes easier to appreciate the membrane's role as a guide rather than a barrier. It does not stop water from moving. It shapes that movement, ensuring that it supports rather than undermines cellular life. This guidance is subtle, yet profoundly important. In physiological systems, osmosis operates across vast numbers of cells simultaneously. Blood vessels, tissues, and organs all rely on coordinated water movement to function properly. The principles remain the same at every scale. As this part gently comes to a close, allow the image of water movement to remain softly present. Picture water molecules drifting across membranes, responding to quiet differences, restoring balance without effort. There is no urgency, no excess, only steady adjustment unfolding in its own time. In the next part of our journey, we will explore how different external environments influence water movement and cell behavior, introducing the concept of tonicity. For now, it is enough to rest with osmosis itself, a process that sustains life through simplicity, patience, and the quiet flow of water toward balance. 15. Tonicity and cellular response. As our understanding of osmosis gently settles, it becomes natural to consider how the surrounding environment influences water movement and, in turn, the behavior of the cell itself. This brings us to the concept of tonicity, a way of describing how the solute concentration outside a cell compares to that inside it. Tonicity does not refer to a substance in isolation, but to the relationship between a cell and its environment, a relationship that quietly determines whether water will enter, leave, or remain balanced. Tonicity is best understood through its effects rather than its definition alone. When a cell is placed in a surrounding solution, water moves according to the relative concentrations of solutes on either side of the membrane. This movement shapes the cell's volume and internal pressure, influencing how it functions. The membrane, once again, serves as the mediator. When the solute concentration outside the cell is similar to that inside, the environment is described as isotonic. In this situation, water molecules move back and forth across the membrane at roughly equal rates. There is no net movement of water in either direction. This balance supports normal cellular activity, allowing processes to unfold without disruption. Many cells in the human body exist in environments that are carefully regulated to be isotonic. Blood plasma and other bodily fluids are maintained within narrow concentration ranges to support cellular health. This regulation occurs at the level of the whole organism, yet its effects are felt at the level of each individual cell. Through this coordination, stability is preserved across scales. When the external environment has a lower solute concentration than the cell's interior, it is described as hypotonic. In this case, water moves into the cell. The influx of water causes the cell to swell as internal volume increases. In animal cells, which lack rigid walls, excessive swelling can become harmful. If water entry continues unchecked, the membrane may stretch beyond its limits. Despite this risk, cells are not helpless in hypotonic environments. They possess mechanisms to regulate internal solute concentrations and adjust transport activity, reducing excessive water influx. These responses unfold quietly, guided by feedback rather than force. The membrane's flexibility and selective permeability play central roles in this adaptation. Plant cells respond differently to hypotonic conditions. Their rigid cell walls provide additional support, allowing them to withstand increased internal pressure. As water enters, pressure builds against the wall, creating a firm state that supports the plant's structure. This pressure is not damaging, but beneficial, contributing to the upright posture of stems and leaves. When the external environment has a higher solute concentration than the cell's interior, it is described as hypertonic. In this situation, water moves out of the cell. As water leaves, the cell shrinks, and internal processes may be disrupted. In animal cells, this shrinkage can interfere with normal function, affecting enzyme activity and structural stability. Once again, cells possess mechanisms to respond. They may increase the uptake of solutes or adjust metabolic activity to restore balance. These adjustments take time, and their success depends on the severity and duration of the external condition. Through gradual response, cells seek to reestablish a workable internal environment. Tonicity highlights the importance of context in biology. The same solution can have different effects depending on the cell involved. A solution that is isotonic for one cell type may be hypotonic or hypertonic for another, depending on internal solute concentrations. This variability reflects the diversity of cellular life and the specificity of biological systems. What remains constant is the principle guiding water movement. Water responds to differences in solute concentration, moving toward balance. The membrane allows this movement while moderating its effects. Through this interaction, cells remain responsive without becoming unstable. In the broader context of physiology, tonicity plays a role in processes such as fluid distribution, blood pressure regulation, and nutrient absorption. At the cellular level, it influences volume, shape, and mechanical stress. These effects ripple outward, connecting microscopic water movement to whole body function. There is something calming about the predictability of these responses, Given a particular environment, the direction of water movement can be anticipated. The cell does not need to decide how to respond. It follows physical principles that unfold reliably, moment by moment. In the context of rest and learning, tonicity offers another gentle metaphor. When external demands are balanced, the internal state remains steady. When demands are too great or too small, adjustment is required. Balance emerges not from rigidity, but from responsiveness, from the ability to adapt without losing coherence. Tonicity also underscores the importance of regulation at multiple levels. Individual cells respond to their immediate environment, while organisms regulate the composition of that environment to support cellular health. This layered regulation creates resilience, allowing life to persist across changing conditions. As we rest with this idea, it becomes easier to see how water movement, solute concentration, and membrane properties work together. None of these elements acts alone. Balance arises from their interaction, guided by simple principles applied continuously. As this part gently comes to a close, imagine a cell resting in its environment, water moving softly across its membrane. In an isotonic state, there is calm exchange without change. In hypotonic or hypertonic states, adjustment begins, guided by gradients and structure. In the next stage of our journey, we will move beyond passive processes and explore forms of transport that require energy input, beginning with active transport. For now, it is enough to rest with tonicity, a concept that reveals how cells sense and respond to their surroundings through the quiet, constant movement of water. 16. Active transport and energy use. As we move gently beyond the quiet processes of passive transport, we arrive at a form of movement that requires a different kind of involvement from the cell. Active transport describes the movement of substances across the cell membrane against their natural gradients, from regions of lower concentration to regions of higher concentration. This kind of movement does not occur spontaneously. It requires energy, careful coordination, and specialized molecular machinery. Yet, even here, the process unfolds calmly, with no sense of strain or urgency. To understand why active transport is necessary, it helps to return briefly to the idea of gradients. Diffusion and osmosis rely on differences in concentration or charge, allowing substances to move toward balance. Over time, these movements tend to eliminate gradients. If cells relied only on passive processes, their internal composition would eventually mirror that of their surroundings. This outcome would make it impossible to maintain the specialized conditions required for life. Active transport allows the cell to preserve and even create gradients. By moving substances against their natural tendencies, the cell maintains internal environments that differ significantly from the outside. These differences support essential functions such as electrical signaling, nutrient accumulation, and waste management. Without active transport, the organized chemistry of the cell would gradually dissolve. Energy for active transport comes primarily from molecules that store and release chemical energy. This energy is not expended recklessly. It is used with precision, driving specific movements that serve clear purposes. Transport proteins involved in active transport are carefully structured to harness this energy, converting it into directed motion across the membrane. These proteins operate through cycles of change. They bind to a substance on one side of the membrane, undergo a shift in shape powered by energy release, and then release the substance on the other side. Once the cycle is complete, the protein returns to its original form, ready to repeat the process. Each cycle is small and contained, yet repeated countless times. It sustains the cell's internal balance. Active transport proteins are highly specific. Each one typically transports a particular type of ion or molecule. This specificity ensures that energy is not wasted moving unnecessary substances and that critical gradients are preserved accurately. Precision, rather than force, defines this process. One of the most important roles of active transport is the maintenance of ion gradients. Ions such as sodium, potassium, calcium, and hydrogen play central roles in cellular physiology. Their uneven distribution across the membrane underlies processes ranging from nerve impulses to muscle contraction and energy production. Active transport ensures that these distributions are established and maintained. Consider the movement of ions that tend naturally to diffuse down their gradients. Active transport works quietly in the opposite direction, restoring differences as they are eroded by passive movement. This continuous balancing act supports stability. Passive transport and active transport do not compete. They complement one another, forming a dynamic system in which gradients are both used and replenished. It is worth noting that active transport does not operate constantly at maximum capacity. Cells regulate transport activity according to need. When gradients are sufficient, transport may slow. When conditions change, activity increases. This regulation conserves energy while preserving responsiveness. In this way, active transport contributes to the cell's adaptability. Cells can respond to changes in nutrient availability, external ion concentration, or internal demand by adjusting transport rates. These adjustments occur through changes in protein activity, expression, or availability, unfolding over time without disruption. The membrane itself provides a stable setting for these processes. Its fluid nature allows transport proteins to function effectively, while its selective permeability ensures that active transport has meaningful effects. Without the membrane's barrier properties, actively transported substances would simply diffuse away. From a broader perspective, active transport illustrates how life balances effort and ease. Where nature provides a path through passive movement, the cell follows it. Where balance requires additional input, the cell invests energy deliberately. This selective use of energy reflects an economy of action, supporting sustainability at every level. There is something reassuring about the restraint inherent in this system. Each active movement serves a purpose, contributing to the maintenance of internal conditions that support life. Even in complexity, there is calm efficiency. In the context of rest and learning, active transport offers another gentle metaphor. Some understanding comes easily, flowing along natural lines of thought. Other insights require effort, repetition, and focused attention. The key is balance, knowing when to allow ideas to drift in and when to invest energy in deeper integration. Active transport also reminds us that boundaries are dynamic. They are not static lines, but active sites of exchange and regulation. The membrane does not merely separate inside from outside. It actively shapes that relationship, preserving difference where difference is needed. As cells age or encounter stress, the efficiency of active transport can change. Maintaining transport systems is essential for long-term cellular health. When these systems falter, gradients weaken, and cellular function declines. This connection underscores the importance of active transport not as an optional feature, but as a foundational requirement for life. Despite its importance, active transport remains largely invisible. It does not announce itself. It simply continues, moment after moment, sustaining conditions that allow all other processes to unfold. This quiet persistence mirrors many of the body's regulatory systems, which operate continuously without conscious awareness. As this part gently draws to a close, allow the idea of energy-driven movement to rest softly in the background. Imagine transport proteins cycling calmly, ions being guided against gradients, and balance being preserved through steady effort. There is no rush, no strain, only purposeful motion unfolding at a measured pace. In the next part of our journey, we will explore a specific and especially important example of active transport, one that plays a central role in nerve and muscle function. For now, it is enough to rest with the broader concept of active transport. a process that sustains life by investing energy where balance requires it, quietly and continuously. 17. The sodium-potassium pump. As we continue deeper into the calm landscape of active transport, one mechanism stands out for its quiet consistency and fundamental importance. This is the sodium-potassium pump, a transport system found in the membranes of nearly all animal cells. It works continuously, without pause or recognition, maintaining conditions that make many other cellular processes possible. Though its actions are small and repetitive, their cumulative effect sustains life at every moment. The sodium-potassium pump is a membrane protein that uses energy to move ions across the cell membrane. Specifically, it transports sodium ions out of the cell and potassium ions into the cell, both against their natural concentration gradients. This movement does not occur by chance. It is carefully coordinated, precise, and rhythmic, forming a steady cycle that repeats thousands of times each second in every active cell. Inside most cells, potassium ions are more concentrated, while sodium ions are less so. Outside the cell, the opposite is true. Left alone, diffusion would gradually erase these differences. Sodium would drift inward, potassium would drift outward, and the internal environment would slowly lose its distinct character. The sodium-potassium pump prevents this outcome, restoring gradients as quickly as they are used. Each cycle of the pump follows a consistent sequence. The protein binds sodium ions from the inside of the cell. Energy is then supplied, triggering a change in the protein's shape. This change carries the sodium ions across the membrane and releases them outside the cell. The protein then binds potassium ions from the external environment. As it returns to its original shape, it releases these ions inside the cell. The cycle then begins again. This process requires energy, typically supplied by a molecule that stores chemical energy. The energy is not released all at once, but used precisely to drive each step of the cycle. The pump does not rush. It moves ions steadily, maintaining balance through repetition rather than force. What makes this pump especially important is the role it plays in maintaining electrical gradients. Because sodium and potassium carry positive charges, their uneven distribution across the membrane creates an electrical difference. This difference is essential for nerve cells, which rely on rapid changes in electrical charge to transmit signals. Muscle cells, too, depend on these gradients to contract and relax. Without the sodium-potassium pump, nerve impulses would fade, muscle contractions would weaken, and coordinated movement would become impossible. Even processes not directly related to signaling would be affected. As ion balance influences water movement, enzyme activity, and cellular structure, despite its significance, the pump does not operate in isolation. It works alongside passive transport processes that allow ions to move down their gradients. Each time sodium enters the cell or potassium leaves through channels, the pump responds by restoring the original distribution. In this way, active and passive transport form a dynamic partnership, each process shaping and supporting the other. The pump's activity also influences cell volume. By regulating ion concentrations, it indirectly controls the movement of water. When ions accumulate inside the cell, water tends to follow. By exporting sodium, the pump helps prevent excessive water entry, protecting the cell from swelling. This protective role is subtle yet vital. Energy investment in the sodium-potassium pump is substantial. In some cells, a significant portion of energy use is devoted to this single mechanism. Yet this investment is not wasteful. It supports stability across countless processes, making it one of the most efficient uses of energy in the cell. It is calming to consider how predictable this system is. The pump does not respond to emotion or urgency. It follows its cycle faithfully, guided by molecular structure and energy availability. Even as conditions change, it continues its work, adjusting only in rate, not in purpose. From an evolutionary perspective, the sodium-potassium pump represents a refined solution to a fundamental problem. Early cells needed a way to maintain internal differences in ion concentration. Over time, this mechanism emerged and proved so effective that it became nearly universal among animal cells. Its widespread presence speaks to its reliability and importance. In the context of rest and learning, the pump offers a gentle metaphor for steady effort. Some forms of balance require continuous investment. They are not maintained by a single action, but by ongoing, consistent work. There is no drama in this effort, only quiet persistence. The pump also illustrates how small actions accumulate into large effects. Each cycle moves only a few ions, yet across trillions of cycles, the impact is profound. This principle appears throughout biology, where life is sustained by repetition rather than intensity. As we reflect on this, it becomes easier to appreciate the body's constant, unseen labor. Even in stillness, even during sleep, cells continue their work. The sodium-potassium pump remains active, preserving the conditions that allow rest itself to occur. Without it, the calm state of sleep would not be possible. The membrane provides the stage on which this process unfolds. Its structure supports the pump's activity, positioning it correctly and maintaining the gradients that give its work meaning. Together, membrane and pump form a system that balances energy use with necessity. As this part gently draws to a close, allow the image of the sodium-potassium pump to settle softly in the mind. Imagine a quiet cycle repeating steadily, ions moving in and out with calm precision, balance being restored again and again. There is no rush, no exhaustion, only continuous, measured activity. In the next part of our journey, we will explore another form of membrane transport that involves larger movements and structural changes, known as vesicular transport. For now, it is enough to rest with the sodium-potassium pump, a quiet guardian of balance whose steady rhythm supports the living cell, moment by moment, without pause. 18 Vesicular transport, endocytosis, and exocytosis. As our exploration continues beyond the movement of individual ions and small molecules, we arrive at a form of transport that operates on a larger scale. Vesicular transport allows cells to move sizable materials across the membrane by reshaping the membrane itself, rather than guiding substances through channels or carriers. The cell encloses them within small membrane-bound structures called vesicles. This process unfolds calmly and methodically, extending the membrane's role from selective passage to gentle containment. Vesicles are small, spherical compartments formed from the membrane. They carry substances either into or out of the cell, depending on the direction of transport. This movement does not involve passage through the membrane in the traditional sense. Instead, the membrane bends, folds, and pinches off, creating a sealed space that travels to its destination. In this way, vesicular transport preserves the membrane's integrity while allowing the movement of materials that would otherwise be excluded. When a cell brings material into itself using vesicles, the process is known as endocytosis. The membrane gradually curves inward around substances outside the cell, enclosing them within a pocket. This pocket deepens, narrows, and eventually separates from the surface, forming a vesicle inside the cell. Once enclosed, the contents can be processed, distributed, or broken down as needed. Endocytosis allows cells to take in nutrients, signaling molecules, and even other cells or particles. Some forms of endocytosis are relatively nonspecific, capturing small volumes of surrounding fluid. Others are highly selective, relying on membrane proteins that recognize and bind particular substances before vesicle formation begins. This selectivity ensures that the cell invests energy only where it is beneficial. The reverse process, exocytosis, moves materials out of the cell. Vesicles formed within the cell travel toward the membrane, guided by internal structures. When a vesicle reaches the membrane, it merges with it, releasing its contents into the external environment. The membrane of the vesicle becomes part of the cell membrane, maintaining continuity as materials exit. Exocytosis is essential for secretion. Cells release hormones, neurotransmitters, enzymes, and other substances through this process. Each release event is controlled, occurring only when conditions are right. In nerve cells, for example, exocytosis allows chemical signals to pass between cells, supporting communication throughout the nervous system. Both endocytosis and exocytosis require energy, shaping the membrane, moving vesicles, and coordinating fusion events all depend on controlled energy use. Yet even here, the process remains measured rather than forceful. Vesicular transport unfolds through a series of small, coordinated steps, each guided by molecular interactions. The membrane's fluid nature is crucial for vesicular transport. Without flexibility, it could not bend or fuse as required. Phospholipids and proteins shift position, accommodating changes in curvature without compromising the membrane's barrier function. This adaptability allows the membrane to serve as both boundary and transport mechanism. Vesicular transport also plays a role in membrane renewal. As vesicles fuse with the membrane during exocytosis, they add new material. During endocytosis, portions of the membrane are removed. Through these opposing processes, the cell maintains membrane composition and surface area, adjusting to growth, division, or environmental change. Within the cell, vesicles do not move randomly. They follow organized pathways, guided by internal structures that ensure they reach the correct destination. Some vesicles deliver materials for digestion, others transport newly synthesized proteins, and still others prepare substances for secretion. This organization allows the cell to manage complexity without confusion. From a broader perspective, vesicular transport allows cells to interact with their environment in richer ways. They are not limited to exchanging small molecules. They can ingest complex materials, respond to signals, and release substances that influence distant cells. Through vesicles, the membrane becomes an active participant in cellular dialogue. It is soothing to reflect on how seamlessly this process integrates with other forms of transport. Channels, carriers, pumps, and vesicles all contribute to exchange, each operating within its own scale and context. Together, they form a comprehensive system that supports life through balance and coordination. Vesicular transport also illustrates how structure and function are intertwined. The ability to form vesicles depends on membrane composition, protein interactions, and energy availability. Each element supports the others, creating a system that is resilient and adaptable. In the context of rest and learning, vesicular transport offers another gentle metaphor. Some ideas are small and pass easily into awareness, like ions through channels. Others are larger and require time, preparation, and integration. They arrive wrapped in context, gradually unfolding within the mind. Both forms of learning have their place, contributing to understanding in different ways. Even in quiet moments, vesicular transport continues. Cells constantly adjust their surfaces, recycle components, and manage resources. This activity does not disturb the overall calm. Instead, it supports stability, ensuring that materials are where they need to be. As we near the end of this part, imagine the membrane once more, gently curving and reshaping itself. Vesicles form, travel, and merge, releasing or enclosing substances with care. There is no abrupt motion, only steady transformation, guided by structure and energy. In the next stage of our journey, we will step back and connect these transport processes to everyday physiological functions, exploring how membrane transport supports the living body as a whole. For now, it is enough to rest with vesicular transport itself, a process that expands the membrane's role from selective gatekeeper to graceful carrier, sustaining life through quiet movement and gentle change. 19. Membrane transport in the living body. As we gently step back from the microscopic details of membranes and transport mechanisms, it becomes possible to see how these quiet processes extend far beyond individual cells. Every movement across a membrane, every ion shifted or molecule enclosed, contributes to the functioning of tissues, organs, and the body as a whole. Membrane transport is not confined to textbooks or diagrams. It is happening continuously within you, sustaining balance, coordination, and life itself. In the human body, trillions of cells rely on membrane transport to meet their needs. Each cell exists within a shared internal environment, yet maintains its own distinct internal conditions. This harmony between individuality and cooperation depends entirely on the regulated movement of substances across membranes. Without it, the body would quickly lose its ability to function as an integrated whole. Consider the simple act of breathing. When air enters the lungs, oxygen fills tiny sacs lined with delicate cells. Oxygen molecules diffuse across these cell membranes into the bloodstream, driven by concentration differences. At the same time, carbon dioxide diffuses in the opposite direction, leaving the blood to be exhaled. These exchanges occur effortlessly, guided by diffusion and membrane permeability, repeating with every breath. Once oxygen enters the blood, it is carried to tissues throughout the body. There, it diffuses once again across cell membranes, entering cells that require it for energy production. Inside these cells, oxygen supports the release of energy that powers active transport, synthesis, and repair. In this way, membrane transport links breathing to cellular metabolism, connecting the external environment to the deepest levels of life. The digestive system provides another example of membrane transport in action. Nutrients broken down from food must cross the membranes of intestinal cells to enter the bloodstream. Sugars, amino acids, ions, and water all move through carefully regulated pathways. Some diffuse passively, others rely on carrier proteins, and still others are actively transported against gradients. This selective absorption ensures that the body receives what it needs while excluding harmful substances. Once nutrients enter the circulation, they are delivered to cells throughout the body. Each cell regulates uptake according to its needs, adjusting transport protein activity as conditions change. A muscle cell during activity may increase glucose uptake, while a resting cell may take in less. These adjustments occur quietly, guided by signaling pathways and membrane responsiveness. The nervous system depends especially heavily on membrane transport. Nerve cells communicate through rapid changes in electrical charge across their membranes. These changes arise from the controlled movement of ions through channels and pumps. Sodium and potassium gradients, maintained by active transport, allow nerve impulses to travel swiftly and reliably. Each thought, sensation, or movement depends on these ionic exchanges occurring precisely when and where they are needed. Muscle contraction, too, relies on membrane transport. Ions move across membranes within muscle cells, triggering the interactions that lead to contraction and relaxation. Calcium ions play a central role, entering and leaving compartments in a carefully timed sequence. The membrane systems that manage this movement ensure smooth, coordinated motion rather than chaos. In the circulatory system, membrane transport supports the regulation of fluid balance and blood pressure. Water and ions move across the membranes of blood vessel cells, adjusting vessel tone and volume. These processes help maintain stable circulation, ensuring that tissues receive a consistent supply of oxygen and nutrients. The kidneys provide a particularly clear illustration of membrane transport on a large scale. These organs filter blood, selectively reabsorbing needed substances while excreting waste. Along the kidney's filtering units, cells use a combination of passive and active transport to fine-tune the composition of bodily fluids. Sodium, potassium, water, and other solutes are moved across membranes with remarkable precision. Hormonal signaling also depends on membrane transport. Hormones, released by glands, travel through the bloodstream and interact with target cells. Some hormones cross membranes directly, while others bind to receptors on the cell surface. In both cases, the membrane serves as the point of contact, translating chemical messages into cellular responses that influence growth, metabolism, and behavior. The immune system relies on membrane transport and recognition to protect the body. Immune cells move through tissues, responding to signals and interacting with other cells through membrane-bound receptors. Vesicular transport allows immune cells to engulf pathogens, process them, and present fragments for recognition. These activities depend on membranes that are flexible, responsive, and precisely regulated. Even the maintenance of body temperature involves membrane transport. Cells adjust ion movement and metabolic activity in response to temperature changes, helping generate or conserve heat. At the surface of the skin, membrane transport influences sweating and blood flow, contributing to thermal regulation. What is striking about all these examples is how little conscious effort they require. Instead, it relies on cellular systems that respond automatically to conditions. Membrane transport mechanisms operate continuously, adjusting to internal and external changes without interrupting overall function. This quiet autonomy allows the body to remain stable even as circumstances vary. Whether at rest or in motion, asleep or awake, cells continue their work. Membranes regulate exchange, gradients are maintained, and balance is preserved. The constancy of these processes provides a foundation upon which all other activities depend. In moments of rest, such as sleep, membrane transport remains active. Cells repair damage, restore gradients, and prepare for the next period of activity. The calm state of sleep itself is supported by transport processes that regulate brain chemistry and neuronal signaling. Without these mechanisms, rest would not be restorative. There is a deep sense of reassurance in knowing that life is sustained by such steady, reliable processes. Membrane transport does not seek attention. It does not announce success or failure. It simply continues, moment after moment, supporting the body's needs with quiet precision. In the context of learning, this perspective can feel grounding. Understanding the body at this level reveals not fragility, but resilience. Balance is maintained not by perfection, but by continuous adjustment. Small shifts are corrected before they become large problems, allowing stability to persist. As this part gently draws to a close, imagine the countless membranes throughout the body, each one regulating exchange in its own small domain. Picture ions moving, water flowing, nutrients entering, and wastes leaving, all guided by the same principles we have explored. Together, these movements sustain the living body, connecting the microscopic to the whole in a seamless, continuous flow. In the final part of this session, we will step back once more, reflecting on the membrane as a quiet guardian of balance and drawing together the ideas we have explored. For now, it is enough to rest with the living body itself, supported at every moment by the calm, unceasing work of membrane transport. 20. The membrane as a quiet guardian of balance. As we arrive at the final moments of this long, gentle exploration, it is helpful to return to the image that has guided us throughout. The cell membrane, thin and unobtrusive, surrounds every living cell, quietly defining what it means to be alive. It does not draw attention to itself, yet it supports every process we have considered. In this closing reflection, the membrane can be seen not as a collection of parts, but as a steady presence, a guardian of balance whose work never truly stops. From the earliest moments of life on Earth, membranes have played this role. Long before complex organisms appeared, simple cells relied on boundaries to maintain internal order. Those early membranes separated chemistry from chaos creating protected spaces where reactions could proceed reliably. Over vast stretches of time, this simple solution proved so effective that it became a defining feature of life itself. Throughout our journey, we have seen how the membrane balances openness with protection. It allows oxygen to drift inward and carbon dioxide to leave, supporting respiration without effort. It guides water gently across its surface, preserving volume and internal pressure. It opens channels for ions, carries nutrients through specialized proteins, and invests energy where gradients must be preserved. At every step, it maintains equilibrium without rigidity. What is remarkable is not just what the membrane does, but how it does it. There is no central command, no conscious oversight. Instead, structure and physics work together. Phospholipids align, proteins shift shape, and gradients form and dissolve according to simple rules. From these interactions, stability emerges. The membrane does not resist nature. It works with it, shaping natural tendencies into life-sustaining patterns. In this way, the membrane embodies a broader principle found throughout biology. Life persists not by opposing the world, but by adapting to it. Boundaries do not isolate. They enable meaningful interaction. Selectivity does not restrict life. It makes it possible. The membrane's role as a guardian of balance is perhaps most evident in times of change. When conditions outside the cell shift, the membrane responds. Transport rates adjust, proteins reorganize, and gradients are restored. These responses are gradual rather than abrupt, preserving continuity even in the face of fluctuation. This steadiness allows cells to endure stress, adapt to new conditions, and recover from disruption. At the level of the whole organism, the combined work of countless membranes creates harmony. Organs coordinate their functions, tissues maintain their structure, and systems communicate seamlessly. Each cell contributes to this larger balance by managing its own internal environment. The membrane is the point where individuality and cooperation meet. It is comforting to realize that this balance continues even during rest. While the conscious mind slows, cells remain active. Membranes continue to regulate exchange, restore gradients, and prepare for future activity. Sleep itself is supported by these processes, as neurons adjust their chemistry and energy stores are replenished. The calm of rest is mirrored by the steady work unfolding at the cellular level. As listeners, there is no need to carry every detail forward. Understanding, like membrane transport, does not require holding on to everything at once. What matters is the overall sense of harmony, the recognition that life is sustained by quiet, continuous regulation rather than dramatic intervention. The membrane reminds us that boundaries are not barriers to peace. They are conditions for it. By defining limits, the membrane allows stability to exist within change. This idea extends beyond biology, offering a gentle reflection on balance in many aspects of life. In this final moment, imagine once more the living surface of a cell. Visualize proteins moving softly, channels opening and closing, vesicles forming and merging. All of this occurs without sound, without haste. Balance is maintained not through force. Across trillions of cells, this same pattern repeats. Each membrane performs its role faithfully, contributing to the larger whole. Together, they sustain breathing, movement, thought, and rest. They support life not by standing out, but by remaining steady. As this session comes to an end, there is nothing left to do or remember deliberately. The ideas can settle naturally, like molecules drifting toward equilibrium. Just as the membrane allows what is needed and lets the rest pass by, the mind can release effort and remain at ease. In the quiet that follows, the body continues its work. Membranes persist as they always have, guardians of balance, maintaining the conditions for life with calm reliability. And in this understanding, there is a sense of completion. a gentle closing, as the steady rhythm of cellular life continues on, uninterrupted, supporting rest, renewal, and peaceful sleep.